Hi there, and welcome to this video on A-Level Biology for the AQA specification, focusing on the topic of cells, and in particular on binary fission and viral replication. I'm Manisha from StudyMind, where we help you to revise A-Level Biology with our helpful video tutorials tailored to your subject, your specification, and to you. If you're new here, please make sure to click that subscribe button. And whilst you're watching, feel free to leave any comments down below of anything you're unsure about, and let us know if it's your first time watching so we can send you our free revision resources. We also have helpful timestamps to guide you through the specification. Welcome to lesson 6 of 9 in this tutorial, covering binary fission and viral replication. This is the 6th video in our series of 9 lessons on the topic of cell structure. In the last lesson, we looked at the principles of the cell cycle. Here are the key learning objectives for today's lesson. First, we'll be looking at binary fission, and then we'll move on to viral replication. Here are the AQA learning objectives for today's lesson. Feel free to pause the video now and have a quick read through them before we begin. First, we will cover binary fission. Prokaryotes divide by a simple process called binary fission. Prokaryotic cells can replicate their DNA and split into 2 daughter cells that are genetically identical to each other and to the parental cell. Before binary fission, there is circular DNA, or a bacterial chromosome, and plasmids, which are tightly coiled. During DNA replication, the circular DNA uncoils and duplicates. The plasmids may duplicate many times. Next, the cytoplasm extends. It elongates, which separates the 2 sets of DNA. Each end receives 1 bacterial chromosome and equal plasmid amounts. The cytoplasm begins to split, and 1 cell needs to split into 2. This begins with the splitting of the cytoplasm. Cell walls develop through the centre of the cell to begin the splitting process. The new cell wall is fully formed, which eventually splits the cytoplasm into 2. The cytoplasm splits fully now to produce 2 new cells. These daughter cells are genetically identical and have an equal share of the genetic material. Now, we'll be looking at how viruses replicate. Viruses do not undergo cell division. Instead, they infect a host cell and use it to replicate themselves and to produce new viral particles. First, the virus binds to the host cell using its attachment proteins. Viruses can have different attachment proteins, which means that different viruses affect different types of cells. The HIV virus, for example, has attachment proteins which bind to receptors on the surface of human T cells. Once the attachment proteins of a virus attach to the host cell, the virus latches onto the cell and injects the host cell with its nucleic acids, either DNA or RNA. These viral proteins can force the cell to stop making proteins and replicate DNA for the cell. Instead, it will focus all its energy into replicating the viral genome and proteins. Once the host cell has produced a sizable number of viral particles, these particles will burst through the cell through lytic release to infect other host cells. The original host cell is now dead because of a gaping hole in its cell membrane. We've now covered all the learning objectives for today's lesson. Feel free to skip back through the video and re-watch anything you feel unsure about. We've now completed Lesson 6. If you enjoyed this tutorial, make sure to subscribe by clicking down below and leaving a comment of the topic that you'd like to see a video on. Click here to watch the rest of our videos in our A-Level Biology series, or visit our website studymind.co.uk for past paper compilations by topic and specification.